Hi, so today we're going to talk about cells. So the discovery of the cell came with the invention of the microscope. So early microscopes. In 1665, Robert Hooke used an early compound microscope to look at a thin slice of cork, which is a plant material. The cork looked like thousands of tiny empty chambers. He called these chambers cells. And cells became, the definition of cells is that they're the basic units of life. So if we look here, this is this microscope that Robert Hooke used. Very early compound microscope. This was Hooke's drawing of the cork cells. He called these tiny little chambers you see here cells. At the same time, Anton van Leeuwenhoek used a single lens microscope to observe pond water and other things. So this was his microscope. And the microscope revealed a world of tiny living organisms. So from the discovery of cells by these two scientists came cell theory. Cell theory has three parts. 1, all living things are composed of cells. 2, that cells are the basic units of structure and function in living things. And new cells are produced from existing cells. So there's two main types of cells, prokaryotic cells and eukaryotic cells, or prokaryotes and eukaryotes. Cells come in a variety of shapes and sizes, but they have two things in common. No matter if they're a prokaryotic cell or a eukaryotic cell, they are all surrounded by a barrier called a cell membrane. And here's a picture of a cell membrane, the structure of it, and at some point contain DNA. So all cells contain DNA and have a cell membrane. They are classified into two categories depending on whether they contain a nucleus. The nucleus is a large membrane-enclosed structure that contains the cell's genetic material in the form of DNA. So here's your nucleus inside your cell. Here's a picture of the cell. This is the nucleus. Inside it, we have different structures that we'll talk about in the next section, but the biggest part is the DNA. And the nucleus, its function is to control the cell's activities. So eukaryotic cells, or eukaryotes, are cells that contain a nucleus. Prokaryotic cells, or prokaryotes, are cells that do not contain a nucleus. So here's a eukaryotic cell, and here's its nucleus. Here's a prokaryotic cell, and there is no nucleus. The DNA is just floating around in the cytoplasm of the cell. So prokaryotic cells have genetic material that is not contained in the nucleus. They do not have membrane-bound organelles, and they're generally smaller and simpler than eukaryotic cells. An example would be a bacterial cell. They're prokaryotic cells. And here's its cytoplasm. There's no membrane-bound organelles, and its DNA, which is right here, is just floating around inside the cell. Eukaryotic cells, on the other hand, contain a nucleus in which their genetic material is separated from the rest of the cell. So the genetic material, the DNA, is located here inside the nucleus. Eukaryotic cells are generally larger and more complex than prokaryotic cells. They generally contain dozens of structures and internal membranes. Many eukaryotic cells are highly specialized. And plants, animals, fungi, and protists are all examples of eukaryotic cells. So let's delve a little bit deeper into the structure of eukaryotic cells. They are the most complex cell. So they have a lot of different structures. So the structures within a eukaryotic cell that perform important cellular functions are known as organelles. Cell biologists divide the eukaryotic cell into two major parts, the nucleus and the cytoplasm. The cytoplasm is the portion of the cell outside the nucleus. It's usually this jelly-like substance where all the rest of the organelles in the cell are floating. So here's a plant cell and some of the structures that we're going to go over. We're going to go over the nucleolus, which is inside the nucleus. We're going to go over the smooth endoplasmic reticulum, rough endoplasmic reticulum, cell wall, cell membrane, chloroplasts, vacuoles, mitochondrion, Golgi apparatus, ribosomes attached and ribosomes free, the nuclear envelope, and in the animal cell we're going to go over some of the very same structures. Nucleolus, nucleus, nuclear envelope, rough endoplasmic reticulum, centrioles, mitochondrion, Golgi apparatus, ribosomes attached, cell membrane, ribosomes free, smooth endoplasmic reticulum. And we're going to start with the nucleus. The function of the nucleus is it is the control center of the cell. And the structure is it contains nearly all of the cell's DNA, and with it the coded instructions for making proteins and other important molecules. So within the nucleus is the DNA, and the DNA is basically the control molecule of the cell. So here's the structure of the nucleus. It contains chromatin, which we're going to talk about in a minute. It contains the DNA. The nucleolus, the nuclear envelope, which has nuclear pores. So the nucleus is surrounded by a nuclear envelope composed of two membranes. The envelope is dotted with nuclear pores which allow material to move in and out of the nucleus. So this is what surrounds the nucleus, protects the DNA on the inside, but these pores allow for things to enter and leave the nucleus. The granular material in the nucleus is called chromatin. So this is the chromatin. Chromatin consists of DNA bound to protein. And when a cell divides, chromatin condenses to form chromosomes. And chromosomes contain the genetic information that is passed from one generation of cells to the next. So if we think about chromatin, I think about it looks more like a bowl of spaghetti. But as we condense it around these proteins called histones, we make chromosomes. So here's a condensed chromosome. We condense it into these chromosome structures. These are then passed on to the next generation of cells. Most nuclei also contain a nucleolus, and the nucleolus is where the assembly of ribosomes begins. So this is where we assemble another structure we're going to talk about, which is ribosomes. Ribosomes, one of the most important jobs carried out in the cell is making proteins, and proteins are assembled on ribosomes. Ribosomes are small particles of RNA and protein found throughout the cytoplasm. So ribosomes produce proteins by following coded instructions that come from the nucleus, and cells that are active in protein synthesis are often packed with ribosomes. So here are the ribosome structure, two subunits. These are made, the ribosome is made in the nucleolus, and it assembles proteins. Okay, so in the cell, there's tons of ribosomes, free ribosomes and attached ribosomes. They're the protein factories. So the endoplasmic reticulum is another structure that contains an internal membrane system called the endoplasmic reticulum, and it is where lipid components of the cell membrane are assembled along with proteins and other materials that are exported from the cell. So its two main functions are to make lipids and to make proteins. This is the endoplasmic reticulum, lots of membranes and infoldings, and on some of the endoplasmic reticulum you're going to see ribosomes. So there are two types, rough and smooth. The portion of the ER involved in protein synthesis is called the rough endoplasmic reticulum, or ER, because that's where proteins are made on ribosomes, so that's where the ribosomes are located on the ER. So ribosomes are found on the surface of the rough ER. The rough ER is abundant in cells that produce large amounts of protein for export. The smooth ER, on the other hand, does not have ribosomes on its surface, and it contains the collections of enzymes that perform specialized tasks, such as the synthesis of membrane lipids and detoxification of drugs. So outside of the nucleus is the rough ER, where proteins and lipids are made, and the smooth endoplasmic reticulum is more commonly used for making lipids, but also for detoxification of drugs. If you look at the liver cells, your liver cells are full of smooth endoplasmic reticulum because that is where alcohol and other drugs are broken down. Next, we have the Golgi apparatus. After the proteins are produced in the ER and on the ribosomes, they move into the Golgi apparatus. It appears as a stack of closely opposed membranes. This is what it looks like. And the function is it modifies, sorts, and packages proteins and other materials from the ER for storage in the cell or secretion for outside of the cell. It's basically like the UPS of the cell. So after the proteins are made on the rough ER, after the lipids are made in the smooth ER, they are transported to the Golgi apparatus, which will package them and transport them where they need to go. From the Golgi apparatus, proteins are then shipped to their final destinations throughout the cell or outside of the cell. Lysosomes are another structure. They're small organelles filled with enzymes. They're the cleanup organelle. They break down lipids, carbs, proteins, and smaller molecules that can be used by the cell. And they also break down organelles that have outlived their usefulness. They also break down bacteria that happen to enter the cell. So basically, they're the cleanup crew. They contain all the enzymes that break everything down. Vacuoles. Some cells, not all cells, contain sac-like structures called vacuoles. Vacuoles are the storage organelle. They store materials such as water, salts, proteins, and carbs. In many plant cells, there's a single large central vacuole filled with liquid. It actually takes up most of the inside of the cell. The pressure of the central vacuole allows the plants to support heavy structures such as leaves and flowers. There's also vacuoles found in some unicellular organisms and some animals. The paramecium right here has what we call a contractile vacuole that pumps excess water out of the cell. So it's like a water pump. If too much water gets inside the cell, it pumps it back out. Mitochondria. Nearly all eukaryotic cells contain mitochondria. The mitochondria are the energy organelle. They convert chemical energy stored in your food into compounds that are more convenient for the cell to use. So this is where your carbohydrates are broken down into actual energy molecules that the cell can use. Mitochondria are enclosed by two membranes, an outer membrane and an inner membrane, outer membrane and the inner membrane. The inner membrane is folded up inside the organelle. Chloroplasts are only found in plants and some other organisms. Chloroplasts capture energy from sunlight and convert it into chemical energy in a process called photosynthesis. So this is the light capturing organelle, and this is the site of photosynthesis. Chloroplasts are surrounded by two membranes, and they contain the green pigment chlorophyll. So there's two membranes, outer and inner, and there's also these stack of thylakoids that we'll talk about later when we talk about photosynthesis. But inside these disc-like structures is where the green chlorophyll is located. Cytoskeleton. Eukaryotic cells are given their shape and internal organization by the cytoskeleton. Just like your cytoskeleton, it's basically helps the cell to maintain its shape, and it's also involved in movement. So that's what your skeleton does as well. But in this case, a cytoskeleton is a network of protein filaments that does this. It's made up of two main filaments, microfilaments and microtubules. So the cytoskeleton is a network of protein filaments that help to maintain its shape. It's also involved in movement. Microfilaments and microtubules, we're going to talk about both. In this case, microtubules are shown in red here. Microfilaments are shown in green. So if we break it down, cell membrane on either side, here are some organelles. Within that structure and kind of going through the cell are your microtubules, which are larger than your microfilaments. But they help keep the structure of the cell, so your cell isn't flat like a pancake and there's no room for your organelles to function. So microfilaments are thread-like structures made up of the protein called actin. They form extensive networks in some cells. And their function is to produce a tough, flexible framework that supports the cell and helps some cells move. Microtubules, on the other hand, are hollow structures made up of proteins known as tubulins. Their function is to maintain the cell shape, and they're also important in cell division, which you guys will see later on in the year. They also build projections from the cell surface, cilia and flagella, that enable some cells to swim rapidly through liquids. And last but not least, in animal cells only, there are structures known as centrioles that are formed from tubulin, which is microtubules. And they're located near the nucleus, and we think they help to organize cell division. The thing is scientists have actually removed the centrioles, and the animal cell can still divide without the centrioles, but they seem to be involved in cell division. And that is all for today. Thanks for watching.